We introduced the concept of the blood-brain barrier last time we spoke, and so we'll go on to the actual structures that form the blood-brain barrier and the importance of that. So before we do so, let's speak about the blood supply to the nervous system. Now, although the brain and spinal cord and our nerves is only about 3 to 4 pounds, so about 2% of our total body weight, the brain and associated structures receive quite a bit of our cardiac output. So about 15% of our total blood supply goes to the brain and spinal cord and nerves. So although it doesn't weigh a whole lot and it doesn't represent a lot in terms of the size of those structures, it does get a significant chunk of our blood supply. And that's because of its high metabolic rate. So the brain uses about 20% of oxygen at rest, and about 50% of the glucose that we consumed, again, at rest, goes to the brain for brain function. And that is an indicator to us of the importance of these functions, right? The high metabolic rate is telling us how important these brain functions are. So it depends on flow for energy. So the brain is very susceptible to lack of glucose and to lack of oxygen or lack of blood supply. And we'll see what are the consequences if that does happen. So neurons rely on aerobic glycolysis exclusively, so they must have a steady supply of glucose, they must have a steady supply of oxygen. And although we talked about a little bit of glycogen being able to be stored in glial cells, there isn't a whole lot of glycogen stores in the brain in general, right? There is a small ability through glial cells, but overall there must be a steady supply of glucose coming in to supply the brain, as well as oxygen. Fatty acids can be converted to ketones, but only in extreme conditions through another pathway. But for the most part, fatty acids are not a reliable, steady source of energy. And when we talk about interruptions in flow, interruptions in oxygen, we usually refer to what's called a stroke. And a stroke is anytime there is a blockage or a decrease in flow to the brain. This can occur for two reasons. Either there is an occlusion in a blood vessel, a cerebral blood vessel. So that's usually what we call an occlusive stroke. Or there can be a hemorrhagic stroke where there's a bleed in one of those blood vessels. So we can have two causes for interruption in flow, either a blockage through plaque in one of those vessels, or a tear in one of those vessels, or injury which results in bleeding, and we call that a hemorrhagic stroke. And what are the results from a stroke? I'm sure many of you have been exposed or have heard or seen a stroke. What are some of the sequelae, the consequences? Mm-hmm. Yep, so we can get droopiness or paralysis, numbness, weakness, usually on one side of the body, just because if we have interruption to flow on one side of the brain, the corresponding side of the body, which is the alternate side, is what is affected. We can also see inability to speak, so speech interruptions, all types of different presentations, numbness and weakness throughout different parts of the body, depending on where in the brain we had that interruption. Okay, so now we'll speak about the blood-brain barrier. So this really important junction between our systemic circulation and blood and our nervous tissue and neurons and so on. And this is formed by capillaries. So it's the site of exchange between the blood and the interstitial fluid. And we've got this single layer of endothelial cells that are really adhered to one another very tightly by tight junctions. And so this limits the amount of substances or particles that can move freely from the blood into the brain, and only substances that can diffuse readily. So think about gases such as oxygen and carbon dioxide, and think about hydrophobic substances such as fats that can move across that lipid bilayer. Those are the only types of molecules And a few other exceptions, such as alcohol. Alcohol can actually move right across the blood-brain barrier. That's why it has that effect on the brain. But for the most part, the brain is very selectively permeable about what can get across this barrier, this membrane. And so the special anatomy is formed really by the capillaries and the tight junctions between those capillaries, as well as the astrocytes. We mentioned the astrocytes, one of our glial cells last time. The star-shaped cells that have those extensions or foot processes that also play a role in forming the blood-brain barrier. Okay, so what we can see here is a traditional endothelial cell on the left. This is what we find in most of the capillaries around our body. And then on the right, we can see the anatomy of the blood-brain barrier. So we've got this endothelial cell. We've got tighter connections between these cells. If you compare the endothelial cell of a traditional capillary on the left, we can see some spaces here or some pores. In the blood-brain barrier, we do not have those pores. We do not have those spaces. We've got really tight junctions or tight connections that prevent any paracellular transport. We've also got the astrocytes with the foot processes. Now, the foot processes of these astrocytes are not actually forming a functional barrier, but what they're doing is they're promoting those tight junctions. So the foot processes themselves are not a barrier, but what they're doing is they're stimulating very tight connections between the endothelial cells. So together, the endothelial cells and the astrocyte foot processes are preventing any movement of substances from the blood out into the interstitial fluid of the brain and eventually the brain tissue. So the types of substances that can go through, again, are anything that can be transported on a carrier. So think about glucose, right? Glucose is needed for the brain, so we need a carrier to get glucose across. We can also move important amino acids that we need for protein building in the brain. We can also move choline across, but those must be transported using a carrier. And then, as I mentioned earlier, we can diffuse certain substances like gases, carbon dioxide and oxygen can readily diffuse, but our larger molecules, blood cells, proteins, carbohydrates, they cannot go through this membrane, and so they're going to be confined to the blood. And what did we say was the purpose of this blood-brain barrier? What was the big purpose of having this special extra layer of protection of the brain tissue? So this was protective, right? This was a protective mechanism to protect the sensitive nervous tissue from any pathogens, from any toxins, drugs that can be circulating in the blood, right? There's only one other place that we see a barrier like this, and that is in the testis. And that is another really highly protected area to protect the reproductive function. Okay, we'll talk about that in the reproductive system.